Thank you for coming. It's my pleasure to introduce our speaker today for the Jones Seminar. We have Jessie Chi Zhang here, who is a postdoctoral research associate at the Beckman Institute at the University of Illinois Urbana-Champaign. As he describes in his bio, he's dedicated his career to developing innovative optical imaging technologies to answer important biological questions and enable better diagnosis and treatment of human diseases. He started off his academic career doing his bachelor's and master's in optoelectronics in Tianjin University in China. He then came to Michigan, where he did his PhD in the University of Michigan, Ann Arbor, in the Department of Physical Chemistry, with a research emphasis on surface optical spectroscopy. He then took a postdoctoral position at Purdue University, Weldon School of Biomedical Engineering, and transitioned with his advisor to Boston University Photonics Center in Boston as a postdoc focusing on coherent Raman spectroscopy and microscopy for biomedical applications. He's now currently working at the Beckman Institute at the University of Urbana-Champaign. He has published 46 papers in his career, with several more in submission and review, a large number of conference proceedings, number of awards, and really has had a fantastic career for such a young scientist already. So it's really a pleasure to have him here and to talk about label-free chemical imaging for biology and medicine. Thank you.
Hey, thank you very much, Dr. Poe, for the introduction. So, it's my great pleasure to be here. So thank you all for coming here for my talk. So can you hear me clearly? Okay, no problem. Okay. So thank you. I also want to thank the Thayer School of Engineering Search Committee for inviting me to come here for my presentation. So my topic today is label-free chemical imaging for biology and medicine. So I'm going to talk about something about label-free technology and to get chemical information out of biological samples. So I'm from the University of Illinois Urbana-Champaign. So it's about 1,000 miles away from here. So although it's far away, but I think we share a lot in common with Dartmouth. And first of all, we are all great schools and with great engineering programs. In addition, when I got here, I realized that we're all kind of isolated. So this is the map of our college town. You can see that we're about 2 hours away from big cities. This is the Champaign. But the difference is that Dartmouth is in the middle of the forest, but we are in the middle of cornfields. So if you go there, you'll see cornfields everywhere, like this. And the building that where our lab is located is this Beckman Institute. It's endowed by a great entrepreneur and inventor, Arnold Beckman, and built in 1983. So our mission is a better world through collaborative innovation. And this is another similarity. I think when I came here, I realized Dartmouth is actually a bigger version of our Beckman Institute for collaborative research. So before I started my work at UIUC, I also worked at Purdue University Biomedical Engineering and the Department of Chemistry, and also BU. So actually, these two are the same place, same lab. It's just our lab moved from Purdue to BU. So this transition helped me to become an expert in lab relocation and optics packing, and also the lab reconstruction. You can see a lot of, we built 3 labs. I built 3 labs in the 3 places, and a lot of optics here. So my major expertise is in the field of optics and photonics, but I also work extensively in the field of biology and medical research and chemistry. So my research is actually right there at the intersection of the 3 fields. I consider myself about 45% of an optical engineer and 30% of biologist and also 25% of a chemist. So just some rough numbers to summarize myself. So my research mission is to bridge optical engineering and biomedical sciences to build better connections and improve human health. So more specifically, it can be classified into 3 parts. The first is development of optical imaging, microscopy, and spectroscopy technologies. And the second one is to make scientific discoveries using these technologies in the field of biomedical research. And the third one is towards clinical translations. for better diagnosis and better treatment of human diseases. So I will cover all the three parts in today's presentation, but I'll first of all want to give a brief introduction about what is the label-free chemical imaging and what is Coherent Raman and what is SRS. So then I will talk about my contributions to the field in technological innovations and applications in biomedical science, majorly cell biology, and then towards clinical translation, some of our current work and future emphasis in clinical translations. So, okay, background first. So you might have seen this kind of images in many places. So this is a typical phase contrast image from a phase contrast microscope of living cells. So you see the very beautiful structural information and morphological information, but we don't have any chemical information in it. So chemical information is very critical for us to understand the biological functions and also for diagnostic purposes. So the way to get chemical information, usually we need to use chemical labeling. For example, take a fluorescence as example. So we need to use some kind of fluorophores to let it go to the targets, and then we can see them. If this is not very specific, we can conjugate that to an antibody or embed them into a nanoparticle to make the targeting more specific. This is a wonderful technology, okay, but it has some limitations. The major limitations are this usually relies on the delivery of these chemical labels, and sometimes the delivery can be very difficult. Another possible issue is that they have non-specific binding, so it means they can bind to some unwanted receptors that give us some false information. In addition, many of them cannot be used for living systems. Even if they can be used, they can be toxic and perturb the biological functions. So in the end, many molecules are very difficult to label using this way. For example, the metabolites, the small molecules, it's very difficult to label specifically using fluorescence. Another way to do this chemical imaging is to modify the genomes of the organisms and cells and let the protein we want to see fluorescent, conjugate the GFP to it, but this can only study proteins, and also apparently we cannot apply this to humans for diagnostic purposes. So we want to use a different route. We will use a different method, is using photons to directly get the chemical information. And how can we do that? We use the vibrations of the molecules. The molecules are vibrating like this, and if we shine lasers to them, they will give us the spectrum. So these are called vibrational spectrum. So these are very unique spectral signatures here and can be considered as fingerprints of the molecules for the identification. So one of the most widely used vibrational spectroscopy is based on Raman scattering. So this is Dr. C.B. Raman, and discovered this phenomenon in the 1920s, and Raman scattering was awarded Nobel Prize for Physics in the 1930s. So what it tells us is light can exchange energy with the incoming photons. So it can be shifted to the frequency equals to the original frequency plus or minus the vibrational frequency. So in other words, it tells us light carries information of molecular vibrations. So we only need a laser to interrogate the sample and then collect the scattered photons at different frequencies, so we know what the molecules are in the sample. So this is wonderful, right? We don't need chemical labeling at all. But it has a very it has a drawback. It's a very weak process. So only one photon in every 10 billion photons will undergo Raman transition. So how weak is that? You can consider this equals to find one person named Raman in the whole world population. So it's very weak. It's very difficult to find. So for an image like this, if you use spontaneous Raman spectroscopy to get an image like this, you probably will expect at least half a day for the acquisition. So this means that this technology is not fast enough to study living samples. So how can we improve the speed of Raman scattering so that this label-free technology can be as fast as fluorescence for sample imaging? So Coherent Raman scattering solves this problem. Now, instead of using one laser beam, we need two laser beams. One is called pump, and the other is called Stokes beams. One of the beams needs to be tunable in the frequency domain. So we tune the laser beams until the energy difference between the two beams matches the vibrational energy of the molecule, there will be a strong coupling of the laser energy into the vibrational energy or vibrations. So this will generate a strong signal at the anti-Stokes wavelengths called CARS. So this wavelength is equal to the pump frequency plus the vibrational frequency. At the same time, the energy coupling between the two beams will become very significant. So we'll see a decrease in the pump beam and an increase in the Stokes beam. And that is actually caused by the stimulated Raman process called SRS. So CARS is a new frequency generated, and SRS is basically energy transfer between the two beams. Just keep in mind that part. So both of these technologies, these processes can give us much more signal. So the signal enhancement can be million times. This table compares the spontaneous Raman and coherent Raman in terms of speed and imaging in spectroscopy and laser power. You can see the spontaneous Raman, stimulated Raman and CARS have much faster spectral acquisition speed and imaging speed, even with a much lower excitation power. And another advantage of these coherent Raman technologies is that they don't have a fluorescence background. So here you can see that actually CARS has some background called non-resonance. It's sometimes mixed with the signal and contaminate the image, but SRS is absent of such a background. Another advantage of SRS is it can be operated under room light, so it is very attractive for clinical applications. So my work is mostly focused on the SRS part. So how to detect the signal of coherent Raman? So CARS is very easy. It's new photon generated at a new frequency. We can use the filters to block the input frequency and then collect the photons. But SRS is a little bit different. It's an energy transfer. How can we detect the energy transfer? So we use the modulation and demodulation technique as here. So we modulate one beam, for example, the Stokes beam here. So we turn it on and off. And when the beam is on, SRS will happen and it will cause energy transfer. They will decrease the signal in the pump. And when it's off, nothing will happen, no SRS, so signal will remain the same. So you can see that SRS actually transfer the modulation from one beam to the other. And if we use a lock-in amplifier to detect such an energy transfer, we can detect the signal in SRS. So now you have a general idea already to see what is the CARS and SRS and what is the coherent Raman. So I want to move on to talk about my contributions in the technological development part. So you know that we need two frequencies to excite a single Raman transition, but sometimes this is not enough. I would say in most of the time this is not enough because biological samples are very complex. Usually there are mixtures of molecules that have overlapped the Raman peaks like this. It's very difficult to use a single frequency to separate them. So we need a spectral information. We need to know the spectral pattern in order to separate them. So to get a spectral information at each image pixel is called hyperspectral imaging. So the conventional way to do this is we collect a single frequency image first at one frequency and then tune the frequency over a range step by step and collect a stack of image and use the stack to reconstruct the image information of spectrum at each pixel. So this is actually the first problem I encountered when I started hyperspectral imaging. So because biological samples are living samples, they're highly dynamic. So any dynamic movement in the during this stack collection time will distort the spectrum. How can we solve that problem? So we introduced a multiplex excitation scheme. So instead of using two narrowband lasers, now we use one narrowband and one broadband lasers. So we can simultaneously excite multiple Raman transitions and use an array detector to detect the Raman signal in this a stimulated Raman signal in the broadband beam. But here comes another problem. So you know that each channel we need a lock-in amplifier, right? So if we have 32 channels, we probably need 32 lock-in amplifiers, and each one is about $10,000. So it's impractical to do this way. So to solve this problem, our lab invented a chip called a tuned amplifier, and this chip can subtract the signal in the same way as a lock-in amplifier, but it's much more compact and cost-effective. So we can assemble an array of these chips and to pair with our array detector and allow us to detect 200,000 spectra per second. That equals to 5 microseconds per Raman spectrum. That's the current fastest speed ever reported for Raman spectroscopy. So based on this detection scheme and the array detector, we designed a multiplex SRS imaging system. So we have the two laser beam combined here and sent to the sample. We scan the laser beam in one direction and then move the stage in the other direction to collect the image. And then after the sample, the broadband beam was dispersed by a pair of gratings and projected onto the detector for collection. So we solved the problem of, so first of all, we increased the imaging speed 32 times because we have 32 channels. And, more importantly, we actually solved the problem of the spectral motion artifacts caused by the sample dynamics in living cells. So this allows us to do a high-speed hyperspectral imaging of living cells. We can image, for example, living cells here, and then at the same time generate a 32-channel spectral information, and then use that information to separate different chemicals in the sample, and then get a chemical map like this. And use the chemical map, we can do quantitative analysis and then see how the chemical compositions change in the living cells at a high speed. So this enabled high-content single-cell analysis. We can analyze hundreds of parameters over these chemical contents in a label-free manner. And it tells us a lot of information about cell metabolism. For example, just take one line here. This line tells us in stress condition, these lipid droplets in the cells tend to move to have a broader distribution and tend to move to the edges of the cells. This is an example, so we can dig into more details of all the parameters and make scientific discoveries. For example, one discovery we made is we found how cancer cells change the metabolism to address this starvation condition. So if we starve the cancer cells, the cells actually develop this kind of protrusions here. With our high-content single-cell analysis, we discovered that the lipid droplets also move to these protrusions. And this triggered a systematic study. So the study showed that the droplets move there to release energy and help these protrusions to grow longer and longer, so the cell will become larger and larger. They have a larger surface area. And eventually, they can take more nutrition from the media so they can survive through the starvation. This is a very smart way that cancer cells to deal with the starvation. But with our high-content imaging technology, we can make such kind of discovery. Sorry. So we have improved the hyperspectral imaging speed about 32 times. So we have a throughput about 20 to 30 cells per second. This is not enough. In many cases in single-cell biology, we need to analyze a large amount of cells. And the conventional way to do this is to use flow cytometry. So in flow cytometry, cells flow through the laser focus one by one very rapidly and analyzed by the laser. But we have to sacrifice the spatial information now. In order to get the high speed, we cannot get a spatial information like imaging. We basically take one spectrum for each particle. So the signal can come from light scattering or fluorescence for flow cytometry. So if we want to know the chemical information, we can always label the cells and get the fluorescence signal. But again, fluorescence labeling has the limitations or the potential issues that we mentioned before. Can we use Raman to do this label-free, to do the flow cytometry in a label-free manner? So Raman flow cytometry actually has been demonstrated by a few groups before based on microfluidics. But however, because of the, what happened? Sorry. Because of the slow acquisition speed of the spontaneous Raman spectral collection, so the throughput is on the level of a few cells per second. This is much slower than the fluorescent-based flow cytometry. Since SRS can improve the signal of Raman, can we use SRS to boost the signal? So we designed the first hyperspectral stimulated Raman flow cytometry based on the multiplex excitation and array detection. So we have the cells flow very rapidly here, one by one, and analyzed by the laser. But instead of doing imaging, now we stop the laser at the center of the flowing stream and then collect the spectrum. So you can see we have, using a mixed polymers as example, polymer beads as an example, we can show that we can use this label-free technology to separate different chemical compositions, different particles that have different chemical compositions. Some of them are PMMA, some of the polystyrene. They have different Raman signatures. So we can also compare the SRS signal with the spontaneous Raman signal. We see very good agreement with each other, but keep in mind that we have a million times spectral acquisition speed improvement here. So this allows us to analyze up to 11,000 particles per second. And the PCA analysis can help us to separate different populations. I only show 2 populations here. You can do even more, 3 or more or 4 populations. So using cells as an example, we demonstrated that based on the cell content difference, we can separate them also with the label-free technology, label-free high-speed flow cytometry. So this research delivered a very important message, is that with the help of stimulated Raman, this label-free Raman spectroscopy or Raman technology can be as fast as fluorescence for high-speed single-cell analysis. So it also enabled many potential applications. For example, research has shown that circulating tumor cells has a very different chemical signatures than the blood cells, indicating that we can potentially using the label-free cytometry to detect the circulating tumor cells. Another example is we found that when bacteria develop drug resistance, they tend to have very different chemical signatures than the susceptible ones. So we can also potentially use the technology to study drug resistance in bacteria. So, sorry. I've talked about the multiplex excitation using a narrowband and a broadband lasers. Now I want to move on to talk about some a different excitation geometry. So since the invention of this Coherent Raman technology, almost everybody is using a tightly focused Gaussian beam to do imaging. Because it's the most straightforward geometry for focusing light, and also it has a very high density, energy density at the center of focus. But it has a very important drawback, is that the beam disperses very rapidly after the focus. So we can only tightly focus it into a dot. In order to get 3D information, we need to first of all scan the dot two-dimensionally or move the sample or the focus of the laser in the z direction to get the 3D information. So this may not be the most effective way to do 3D imaging. So we explore a different type of beam. It's called a Bessel beam. So instead of focusing the laser into a dot, the Bessel beam actually focuses the beam into a line and surrounded by a few rings. And this line, also known as central lobe, they can remain focused over a long distance. So this means that the Bessel beam can excite a much larger focal volume. So it has been used in the light sheet microscopy for fluorescence imaging, but it has never been used for Raman or spontaneous, for stimulated Raman. People don't know if it can be used or not. And we show that not only it can be used, it gives us a more, a better way to do volumetric imaging. So we used a pair of hexagons and an objective lens to convert the Gaussian beam to a Bessel beam. And the first figure illustrates the conventional Gaussian beam-based slice-by-slice imaging. So it takes many slides to scan over the 3D volume. The second one shows the Bessel beam-based volumetric imaging. So since the Bessel beam is focused into a line, so a 2D scan of this line can integrate all the information of the 3D volume into a 2D image. This is much faster than the conventional slice-by-slice method. But we don't have the resolution in the z direction now, so it's basically a projection microscopy. But sometimes, if you want to know the overall chemical information in the sample, this is a much faster way. And to get the 3D information, we can do an alternative way. We can use a tomographic approach and getting angle-dependent projection images at different angles and then reconstruct it using mathematical methods. So you may wonder, will the rings of the Bessel beams affect the signal or not? So luckily, the answer is no, because the rings of the two pump and Stokes beam, they do not overlap. And even if they overlap a little bit, because of energy density is very low. So when they multiply them together, the signal is much weaker. So we did a lot of simulation work. And the simulation results show that 98% of the signal is generated in the central loop area. So this is good. So we designed our volumetric imaging system using Bessel beams. And you can see here the two excitons and convert the beam. And we also have a separate beam to bypass these excitons, so we can have a conventional Gaussian-based volumetric image and compare the results. The results show that Gaussian beam basically give us different chemical profiles at different depths, and it takes about 60 frames to sample the entire spectrum, entire volume. And if we add all the 60 frames together, we'll get a global information like this. But the same information or similar information can be obtained with a single 2D scan using the Bessel beam, as we can show here. So we improve the speed about 60 times. For a much larger volume, this is a smaller volume. For a much larger volume, the speed improvement can be even more. And to get a 3D information, we can use a tomographic approach using filter back projection algorithms to reconstruct the image using this angle-dependent projection images. And this is just an example showing the reconstruction results of a 3D structure of chemicals in a large sample. So the capability to image chemical information in 3D is very important because most biological samples are three-dimensional. This can potentially help the diagnostics, such as histopathology, because conventional histopathology, we have to take the sample and then do the sectioning and then staining and 2D imaging. This is not only time-consuming, but also this process can damage the sample and also change the structure. But if we can image them directly with a 2D in 3D with a relatively intact tissue, so we can generate more accurate information for diagnostics purposes. So I've talked about some technological innovations, so I want to move on to continue to introduce some of our current and ongoing projects about biological applications. And the first one we're working on is related to the lipid droplets. So I don't know how many of you have heard of this lipid droplets. So they're actually lipid-rich organelles you can find in most mammalian cells. And these organelles are majorly responsible for the storage and the degradation of lipid droplet, neutral lipids in the cells. You can consider them the harbs of neutral lipids in cells. So what do they look like in the living cells? If you take an image of a Coherent Raman image and tune the lipid frequency on, you can see the image typically like this, and those bright dots are actually lipid droplets. So the majority of the study regarding lipid droplets has been focused on the amount and the content. Very little has been focused on the lipid droplet dynamics. So why? Dynamics is very difficult to study. First of all, it has to be studied in living cells, right? If we fix a cell, no dynamics. And if we put some chemicals there, it can perturb the biological function, and then the dynamic study will not be valid. So conventionally, the droplets are studied using fluorescence. We put some fluorescent dye. For example, this Baldipi is a typical dye to study lipid droplets. We put them in the cells, and using a fluorescence image, you'll see that the bright dots are actually lipid droplets. But if we do image the same field of view with the SRS imaging, we'll see similar contrast because they are all looking at the lipids. But if we look at the cells without adding any labels, without any Baldipi labels, the cells using label-free imaging, the cells actually look like this. You can see that the morphology are very different. The reason is these Baldipi dye is kind of toxic, so they can perturb the biological function significantly. So in this case, the lipid dynamic analysis will not be valid at all if you use conventional methods. So label-free imaging gives us a better way to study lipid droplet dynamics. So this is how we do it. We first of all collect the time-dependent images, and then we use single particle tracking to trace the trajectories and then analyze them statistically. And this is a typical movement of a droplet in a living cell. And using these trajectories, we can define a few parameters to analyze. For example, the maximum displacement from the start to the end, and then the average speed. And you can define many other parameters for the analysis. So typically in living cancer cells, the droplets move like this. You'll see a lot of dynamics in this if you do a label-free imaging. So this tells us a lot of information. So we're trying to decipher that information using quantitative analysis. And using a single particle tracking, we can trace all the trajectories like this. And we found that there are majorly two types of movements. One type is called the active transportation. So that basically these droplets can move to a long distance over a range because they are in the cytoplasma actually. And they can travel along the microtubules and move to different places. They are under a degradation process. The droplets in this case are degradating. So they have a large value in the max D because they're moving a long distance. Another type is called tethered lipid droplets because they are tethered to the endoplasmic reticulum. In this case, they are under a synthesis process. So they are synthesizing there. And they have a much smaller value in the max D. So if the cell metabolism change, this will change the amount of lipids in the synthesis process and the degradation process. These two pools of lipids will have a change, and that will be reflected by the maximum displacement change. That's how we measure this change of lipid metabolism using this dynamic analysis. So let's just give you an example. We can plot the maximum displacement and the speed in a 2D graph like this for a specific cell type in a specific condition. You can consider this is a dynamic nature or dynamic fingerprint or dynamic signature of the cells in a certain condition. And if we change that condition, environment, for example, expose the cells to a lower temperature, we induce some apoptosis or cell death, or we change the environment to create a starvation condition or activate some pathway. I will not get into details of this, but you will see the change. And this change actually tells us how the cell metabolism changed. And it gives us a very rapid way to analyze them because it gives us a very rapid way to analyze the cell metabolism because the lipid dynamics actually change very quickly. It's related to the energy flux of the cells. The content and the amount of lipid droplets do not change very fast because they have a large molecule reservoir to deplete. So that is the reason why we can do a very high-speed detection of metabolism. Not only high speed rapidly, we can also do it very sensitively. This is because we can break the diffraction limit of light because we're using a single particle tracking method. Instead of using conventional microscope resolution, we actually use a single particle tracking similar to those used in super resolution microscopy. So our resolution is about 300 nanometers, but we can trace a very small step movement, much smaller than the 300 nanometers. So this allows us to trace a very small movement step, and we can convert that back to the particle size and correlate that back to the particle size because small particles always move faster. So this allows us to do a much sensitive measurement. For example, in this kind of cancer cells, the lipid droplets are much smaller than the diffraction limit of light. Using the conventional measurement, we can never see the droplets size change after a transition called epithelial to mesenchymal transition because we cannot see them. It's too small. But using a dynamic analysis, we can clearly see the difference or the size change after this transition because we can we can break the diffraction limit of light in this way. Okay, the second project I've been working on is to study the mitochondria. So this is a power plant of the cells, usually majorly responsible for the energy production and also the fatty acid or lipid degradation. So usually they have a lot of proteins, so they are dark in the lipid channel. But we have observed a lot of rod-like structures in the lipid channel. So when we turn on the lipid, we see this. Hyperspectral imaging shows that these rod-like structures have much less lipid content than the lipid droplets, but they have much higher lipid content than something that has a similar shape and size than nearby. So we hypothesize that these are fatty acid-enriched mitochondria. So to get more information, we developed denoising algorithms to convert this image to this, and so we can see a better, higher quality image. And also we use a dynamic or a time-lapse image to trace the trajectory or to trace the movement of these organelles. You can see they moves as an integrity. So they are not the aggregation of different, like a droplets like this. This is the aggregation of droplets. This is actually a integrity, is one organelle. So we further confirm that these are mitochondria using the fluorescence imaging, using mitotracker and also the immunofluorescence. So why this mitochondria start to accumulate lipids? The reason is, we believe, is the hypothermia condition. We expose the cell in the hypothermia condition, basically in the low temperature. Then this will decrease the enzymes in the mitochondria for fatty acid oxidation. So this will mess up the oxidation of lipids. So lipids will accumulate there; they will not be degraded. So if this is true, these fatty acids are very toxic, and the cells need to be find a way to remove them. Otherwise, they will die. So, and we test this by reheating the sample back to the normal temperature. So we first of all create a hypothermia condition at 24 degrees, and then we observe the formation of this lipid-rich mitochondria in the cells. We also see the fission and fusion processes, indicating that they are in a stress condition. So we start to heat the sample at this time point, and we start to observe an interesting thing, that these rod-like structures are converted to dots. You can see pointed out by the red and the blue arrows here. So later, another two rod-like structures also converted to dots. So eventually, all the fatty acid-rich mitochondria were converted to dot-like structures. So these are the movies. You can see. Pay attention to the second one. How this lipid-rich mitochondria were converted to dots here. So this process is we believe is a mitophagy. This is known as how a cancer cell removed mitochondria to remove the malfunctioned mitochondria to reuse them. So what happens here is the hypothermia will create a condition that reduce enzyme activity for the fatty acid oxidation, and then they will create an accumulation of fatty acids in the mitochondria, and then they're toxic, and then the cells start to degrade them after we reheat them back to the normal temperature. So this research shows that the hyperspectral chemical imaging and label-free chemical imaging can help us to make scientific discoveries. And this is the first time that fatty acid accumulation in the mitochondria has been discovered. And also, they give us a lot of potential opportunities. For example, if we can inhibit the mitophagy process, and then the cancer cell will not be able to degrade this mitochondria, then eventually they will die. So this may give us a better way to treat the cancer cells. The last topic, application I'm going to talk about is related to the bacteria. So in recent years, and less and less antibiotics were approved every year. So there's a major problem that the lack of antibiotics will soon become a major health problem. So this is because bacteria has become more and more resistant to antibiotics. So there is a critical need to develop better ways to treat the bacteria infection, or to develop a more sensitive ways to for specific patients to treat the bacteria infection. So the conventional way to test the bacteria antibiotics response is to first of all spread the bacteria on the dish, and then let it grow when the dish is full, and then we start to put the antibiotics compounds here and to let the interaction happen and see the inhibition, measure this called inhibition zone, and measure the interaction area. So this can be very slow for the bacteria species that are obtained from the patients. So especially for some, in many conditions that the concentration of bacteria can be very low, and it takes a long time for it to grow. So we try to see if optical signatures can give us a better way for this antimicrobial susceptibility test. So we focus on something called Pseudomonas aeruginosa. It's a very common bacteria for the urine tract, the respiratory system, soft tissue, and ear infections. So we collect the Carr's coherent Raman signal and also the two-photon autofluorescence signal, you can see that although the Carr's signal looks similar, but the autofluorescence signal decreased a lot after the antibiotics treatment. In addition, we found that if we superposition the Carr's image and the two-photon image, before treatment, the signal do not overlap, and after treatment, they perfectly overlap. So this decrease in autofluorescence signal and also the colocalization of the signal change can be used as an optical signature for improving the antimicrobial susceptibility test. So, but I'm more interested in what molecule gives this signal. So actually, after literature search, we found that it is likely that this molecule called pyoverdine that gave us a signal. And it is the molecule that can give us the color of the bacteria. And it is produced by an enzyme called the PVDA. And we compared the spectrum, absorption spectrum of this molecule with the absorption spectrum of our bacteria strain. We see very similar features here. So also, regarding the location of these molecules, they are circulating around the membrane area. And they also matches what we observed in our images. And for this enzyme, research shown that they form multi-enzyme complexes at the post of the bacteria, also very similar to what we observed here. So we believe what we observed in our two-photon fluorescence is actually from this molecule. And evidence shows that it's very likely. And very interestingly, that you can see here, we observe the bleaching of these optical signals over time. Signal gradually goes away. And at the beginning, we think this is a bad thing. And when you do imaging, the signal goes away, it's always a bad thing, right? Because the absorption of this molecule is at 400 nanometers, and we're using 800 nanometers, so it has a two-photon absorption, so to bleach that molecule. So, and we want to turn this into a positive thing. So, and we think since if this molecule is very important for the bacterial proliferation and also for the biofilm formation, and if we can bleach this molecule, it may give us a better way to treat the bacteria. So if we can use this blue light here, which is absorbed by the molecule, to treat it, and we will be able to kill the bacteria more effectively. So there are many papers targeting this molecule for bacteria removal or control. So we just built a blue light treatment system. We're still testing it, and then we will use it to treat the bacteria to see if it will help to control the bacterial growth. So this project is still ongoing. Our lab designed optical coherence tomography system that used to use light to see the eardrum infection. And this is if the eardrum has some infection and grows some biofilm, we'll be able to see that with this technology. But it is currently a diagnostic tool. So we want to take a step further. Instead of have a diagnostic tool, we want to have the treatment method with it. So we want to couple the blue light with it, and then to give a more powerful diagnostic plus treatment platform. So in the end, I will briefly talk about the translational research. Our lab at UIUC has a lot of expertise and experience in translating optical technology into clinical rooms. For example, we have developed the first version of this retina imaging OCT handheld probe, and also we can change the tip and study the eardrums. And we have the second version, a third version of this one, and become better and better, you can see. And in terms of probe design and also the contrast. And we also developed a briefcase OCT. It's like a very, very, very small. You can carry it with you, and then for the same purpose. So besides OCT to study the eardrum, we also developed the OCT platform to image the tumor margin for breast cancer surgery, and also has built a handheld probe for this tumor margin assessment purposes. So besides the OCT translational research, we're also working on using multimodal imaging, combining multiphoton fluorescence, second harmonic generation, and coherent Raman imaging to study the living, to image the living tissue and get the histology information. So conventional histology, we need to as just mentioned before, we need to fix the sample and do the sectioning and then stain and then image them in two-dimensional. So now, instead of doing this, we can directly take the biopsy and put them on the, our imaging platform and then get a similar or more informative information for diagnostic purposes. So my work actually coupled here, I'm working on to add a Coherent Raman imaging to the system and to make it become more powerful. So this is our ongoing project. Another ongoing project I forgot to list it here is we're actually developing a handheld probe of the same platform, but it's a much smaller probe, so it can be more flexible for the intraoperative diagnosis. So we want to mention one more thing is that one of our future direction is instead of doing conventional biopsy and core needle biopsy, we want to develop a living biopsy system. So we take the core needle biopsy and put it in a microfluidic chamber and to sustain it for a long time for longitudinal study. And this in vitro environment will be able to mimic in vivo condition. We hope to understand more about the in vivo condition using this in vitro platform. We also want to use the fluidic forces to control the position of these samples and build a multimodal imaging system and everything on a portable platform. So that is one of our future directions in clinical translations. So I want to have a brief summary and perspective here. So my work has been mostly focused on the label-free chemical imaging, and it will continue to help the biological research and infiltrate more into the broader applications and discovery of new biological mechanisms. And they can also they will continue to help the medical research and clinical translations and offering us better tools and diagnosis and also discover of new markers and targets for treatment and for diagnosis and treatment. So in addition, the needs in this biological research and medical research will help the optical scientists to develop better tools, including the new modalities and improve the sensitivity, improve the resolution. So I hope in the future I will continue to make progress and make contributions in all the three fields and to push the technology to become more powerful. So the take-home message is you probably will forget like 80% of what I'm talking about today tomorrow. So I hope you to remember just 4 words, the label-free chemical imaging. So that's the take-home message today. So if you remember that in the future, if there's a case that you want to run into this subject or you want to you have a chance to think about it, you can just go Google and search this label-free chemical imaging. You'll find many papers from many groups, including some of contributions from mine. And hopefully you'll realize that this guy came to Dartmouth and talk about it. Okay, so I want to thank my current advisor, Dr. Steve Borpart, for his support of my current research, and Dr. Jixin Cheng for his support of my previous research at Purdue and BU, and all the lab members in current lab and previous lab for the support and help in the past few years. In the end, I want to give my special thanks to Thayer School of Engineering for inviting me, and thank you all for your attention. I'm happy to take any questions you may have.